So, today we are talking about cultivation of bacteria. About physiology of bacteria and specifically about culture media we use for cultivation of bacteria. And as well as you must know, in order to multiply any living organism, both macro and microorganisms, are requiring some environmental conditions. It is related to humans, it is related to animals, to fungi, to bacteria, to everything that lives and multiplies. It requires some environmental factors. You know that if it is cold or if it is too hot, you are not able to perform your daily activities in enough comfort without any troubles. The same thing is related to bacteria. So, whenever we are talking about environmental factors, it is about temperature, it is about pH, it is about oxygen. Sometimes it is required presence of oxygen, sometimes it is required its absence. We'll also talk about that. Mechanical and sonic stress can influence bacteria. Of course, nutrients. All the living organisms are requiring energy and resources for multiplication, because nothing is coming from nothing. Yes. Osmotic pressure is also important. What means osmotic pressure? You must know it from physics, maybe from biophysics, from physiology. In all the living organisms, there are a few types of pressure that are very important. It is osmotic pressure that is provided by what? By electrolytes, by salts, by salts dissolved in liquid. So whenever we are talking about osmotic pressure, we can have three types of osmotic pressure. Or three types of solutions, independence on type of osmotic pressure. First kind of osmotic pressure is hypotonic solution with lack of very small amount of electrolytes dissolved in water. Second one is isotonic solution, is normal amount of electrolytes in liquid. And whenever we are talking about normal, what do we mean by normal? Humans are measuring everything according to what? According to whom? According to us. So we are measuring osmotic pressure in comparison with our inner environment. So inside of our bloodstream, our intercellular space, everything has water, right? And all the time there are some electrolytes dissolved. So normal amount of electrolytes in our liquids, in our physiological liquids, is called isotonic solution and isotonic pressure, osmotic pressure. So everything that is lower of this amount will be called hypotonic solution. Everything that is above, of course, will be called hypertonic solution. This is why physiological solution is also called isotonic solution. Physiological solution is salted water with amount of salt that corresponds to amount of salt in our plasma, in our bloodstream. That's clear, yes? Also, we can have oncotic pressure. And oncotic pressure, so as more amount of the factor that gives pressure is higher is pressure. Oncotic pressure means presence of proteins in liquids. So as higher is oncotic pressure, as higher is amount of proteins dissolved in liquids, as higher is oncotic pressure. Also, we can have actual pressure, for example, pressure in vessels. It is real physical pressure. And this pressure is depending on what? How strong our heart will contract and will pump the blood, and how resistant and elastic are our blood vessels. Because if the heart is not pumping too strong, the pressure will not be too high. If the heart is pumping strongly but our blood vessels are lax, are not too elastic and not too strong, still the pressure will not be high enough, and backwards. It is about pressure. So please make difference between different types of pressure. Yes? Light. And when we are talking about light, some bacteria requires more light and some living organisms are requiring light, because, as well as you know, some organisms are requiring light for what? Photosynthesis. Some organisms are requiring light for their metabolic processes. Or, in presence of light, some metabolic processes will be more efficient. In absence of light, some processes will be less efficient or will pass slowly. One of the very important sides of cultivation of bacteria is bacterial respiration. Not just us as humans, as animals, as plants, as eukaryotes, also prokaryotic microorganisms are requiring respiration. Because if you remember from biochemistry, I guess now you have biochemistry, or you're gonna have it. Do you have it? You have it now. So, very good. If you remember from biochemistry, everything that happens in our cells is not possible without breathing. Because breathing is an exchange of gases that supplies our cells, our body, with oxygen and releases CO2 and extra water. And all of these processes are important for what? For oxidation, for metabolism, for energy production, for reproduction. There is no process in our body that doesn't happen without oxidation and without energy production. So if we are talking about humans, all the time oxidation and energy production, all the respiratory processes are happening just in presence of oxygen. Do you remember from biochemistry what happens if anaerobic respiration happens in our body? Instead of final result of CO2 and H2O or water, final results of metabolism will be lactic acid, right? This is why when somebody is going to the gym and making extra efforts, what happens? not enough oxygen is coming, and in some moments oxidation happens without oxygen, because the physical effort, the mechanical effort is higher than oxygen supply. As a result, in intracellular space will appear lactic acid, and it is not able to get evacuated by bloodstream as water and CO2. It stocks between muscular fibers and produces that painful, that stressful feeling. Because our body is not supposed to pass through anaerobic respiration. So it happens if a little bit of oxygen is missing. But if the oxygen is missing completely, the respiration process is not possible in any cell, and all the cells are starting dying. So basically, any death is related directly or indirectly to respiratory stop. No processes are continuing in the cell, no oxidation happens, no energy production happens, no metabolic processes happens, and the cell is dying, the tissue is dying, the organ is dying, the system is dying, and the body is dying. Same thing happens to bacteria. So if we divide all bacteria in different groups according to their respiration, we can group them in talk about following group: strictly aerobic bacteria with aerobic respiration. What does it mean? Just as us. In presence of oxygen, they are able to live and to multiply. That means if we remove all the oxygen from there, they not just will stop living, multiplying, producing something, consuming something, they will actually die. That's clear. Also, we can have anaerobic respiration with strictly anaerobic bacteria. Anaerobic respiration means all the processes are organized in that way that oxidation and energy production and metabolic processes to happen just in absence of oxygen. So it happens backwards. If we put in there some oxygen, the bacteria will die. They are not able to survive in presence of oxygen. This is why, in order to multiply, to provide proper conditions for anaerobic bacteria, strictly anaerobic bacteria, it is not enough just to give nutrients and just to give necessary moisture and temperature and so on. It is also important to remove oxygen or to supply with oxygen. It depends on type of respiration. Another group of bacteria is called facultative anaerobes. Facultative anaerobes are making part of anaerobic bacteria that are able to modify their metabolism in dependence on circumstances. As a result, the bacteria will get adapted to both aerobic and anaerobic conditions. It has flexible metabolism that is able to change its direction. So, do we reduce oxygen? They will take a break. They will change their metabolic settings, and they will continue multiplying. You supply with oxygen, they take a break, they change their metabolic settings, and they are continuing multiplying. So this process of changing can be stressful for them, but not that dramatic in order to kill them. Also we have one more specific group of bacteria that are called capnophilic bacteria. And capnophilic bacteria, they are also not able to multiply in presence of oxygen, but they require specifically CO2. Capnophilic bacteria are multiplying just in presence of CO2. So in order to let multiplying capnophilic bacteria, it is very important to supply them specifically, not just to remove oxygen, but actually to put their CO2. And if we are talking about fermentation and phototrophic and hematotrophic respiration, it is a kind of special chemical metabolism that is related more not to presence or absence of oxygen or CO2, but mostly to for presence of some specific chemicals that are providing proper bacterial metabolism. And also you can observe in culture media, especially in liquid culture media, how different types of bacteria will act, how they will multiply. So obligate aerobic bacteria, mandatory aerobic bacteria, of course, will try to multiply closer to the surface of the liquid, because it will be closer to the environment, closer to the oxygen. Strictly anaerobic bacteria, backwards, will do its best to get accumulated on the bottom of the tube, because less oxygen is there. Facultative anaerobes will be situated in both places, but basically closer to surface. And aerotolerant anaerobes will be situated homogeneously on the entire in the entire culture media. And there is one more a little bit specific group of microorganisms, microaerophiles. They prefer mostly oxygen, but not too much. It is enough for them just a little bit of oxygen. So they can be misused with anaerobes just because they require a little bit of oxygen in order to multiply. And if we are moving forward to the types of culture media, we can classify them in different ways, in dependence on categories we can apply. For example, if we are talking about their status, their physical form, they can be solid, semi-solid, and liquid. And different culture media, solid, semi-solid, and liquid culture media, are applied for different purposes. You can ask why it is not enough to have just liquid, or just solid, or just semi-solid, just universal. We have different purposes. Sometimes we need to see colonies. If we need to see colonies, it is better to see them on the surface, on the solid surface. Sometimes we need to see just presence or absence of growth. In this case, it doesn't matter too much. Will it be solid or liquid? We'll see growth of bacteria. In some cases, we need to see movement, motility of bacteria. Also, solid and semi-solid culture media will help us better to see it, and so on. If we are talking about its composition and or purpose, we can divide them in two groups: simple and complex culture media. So please don't misuse simple and complex culture media with, for example, simple and complex staining. What is specific about simple and complex staining? Simple staining is staining in which we apply only one dye, yes? and it gives us just a narrow number of features about bacteria. In complex staining, we have more than one dye, more than one component in staining, and it gives us much more features about bacteria. If simple staining is giving us just size, shape, arrangement, that's it. We cannot find anything more about bacteria by applying simple staining. If we are talking about complex staining, it can show us except size, shape and arrangement. It can show us also some specific bacterial structures that we are not able to identify by simple staining. It is about staining methods. When we are talking about cultivation of bacteria, another law is working here. When we are talking about simple culture media, first of all, what is in common? Not too much components. Just as in simple staining, not too much components. If we are talking about meaning and features and targets, simple culture media or common culture media is mostly used for everything. Any kind of bacteria, almost any kind of bacteria can be multiplied, can be cultivated on simple culture media. When we are talking about complex culture media, yes, they have more specific and more complex composition. More different elements will be present in that culture media. But not any bacteria can be cultivated on that culture media. Sometimes even this culture media will inhibit growth of some bacteria in order to help growing other bacteria. So that's clear. Please don't misuse it, because sometimes students are saying simple culture media is for one kind of bacteria, only for one kind of bacteria, and complex culture media for all kinds of bacteria. Doesn't make too much sense. It works backwards, okay? So if we are talking specifically about complex culture media, we have following types. It is one more classification. It is selective culture media, and it is enrichment culture media, it is differential diagnostic culture media, it is special culture media, and it is transport media. Now let's talk a little bit about each of them. Well, as well as you can see, selective culture media helps growing specific types of bacteria. Why it is called selective? Are you selective? At least you should be, right? You should be selective in to whom I am talking, to whom I'm not, with whom I am a friend, with whom I am not. What do I consume? What do I not? It is very important to be selective, because if you will put inside everything, some people are becoming in this way. Where can you put everything? Me, just in trash. So if you respect yourself, you choose, you select what you will introduce in yourself, what you will not, right? If not, you can taste everything in this world. So, same thing. If you're selective, that means not everything is okay for you. When we are and if we're selective, that means we choose from a lot something. That means selective culture media supposes that in sample we take from patient or from the environment, we have too much bacteria. And the mostly common example is intestinal infection. When a person has an intestinal infection, the symptoms will be diarrhea, vomiting, stomach ache, and so on. What do we take as a sample for investigation? Feces, maybe vomiting masses, maybe some rests of food to check, do we have any pathogen there or not? But the mostly common prelevate from patient is feces. We take feces, and what do we have in feces? We have too much bacteria there. Even if the person is not infected more than in intestinal tract, we don't have any other place where we could have more bacteria. On the first place, intestinal tract. On the second place, oral cavity, surprise. And on the last place, our hands. And after that, less and less and less. First three places, intestinal tract, oral cavity, and hands. This is what you should keep in mind. So, even if you are taking feces of healthy person for bacteriological investigation to find there are some bacteria, of course you will find there are a lot of bacteria. Do you need these bacteria? No, you don't. What we are doing, we take selective culture media. Selective culture media will help us to reduce extra bacteria, because looking for bacteria in feces, for specific bacteria in feces, is like looking for a needle in a bunch of needles, actually. So, what do we know about normal flora? Most of normal flora, usually not all of them, but most of them, are gram-positive. What do we know about intestinal infections? Almost all of them are caused by gram-negative bacteria. We take selective culture media. This selective culture media has antibiotic that kills gram-positive bacteria. As a result, from the coproculture, from the sample of stools we put in that culture media, we will not have any gram-positive bacteria. We'll have just gram-negative bacteria. Maybe not just one species, maybe few species, but by the appearance of colonies, we will understand which one is interesting for us. We take sample from that colony. This is how we obtain pure culture from mixed culture. There we'll have few different types of bacteria. We choose one. We have pure culture of bacteria. This is function of selective culture media. Also, we have enrichment culture media. And when you read definition of selective culture media and enrichment culture media, you will observe in both of them something similar. Both selective culture media and enrichment culture media will inhibit growth of unpleasant bacteria and will help growth of bacteria we are looking for. So which is the difference? First of all, most of enrichment culture media are liquid. What does it mean? It supposes we are taking a sample from the place where we are not expecting to find more than one type of bacteria. So if apparently it seems there is no difference between selective and enrichment culture media, basically there is a big difference between them. Because selective culture media we use when we have a sample in which we know for sure we have too much bacteria, we need to choose just one. Enrichment culture media we use when we know for sure we take a sample from the place we'll find just one type of bacteria. But that bacteria is that specific that if we'll have some contamination with normal flora from the environment during cultivation, normal flora will inhibit growth of pathogenic bacteria. As a result, we'll have misdiagnosis. We'll not find the pathogen, we'll find just some of normal flora, and actually we'll find nothing. What is important for us. This is why enrichment culture media will also have some components that will inhibit growth of normal flora in order to let this bacteria that is weak, that is not too much in amount, it will help this bacteria to grow. As a result, we'll see the growth of bacteria and we'll find the pathogen. This is about enrichment culture media. So, selective. We are choosing from a lot something. Enrichment, we take a little bit and we increase it. Just from the name, you can already understand difference between them. Okay? When we are talking about differential culture media, also the name says from itself, differential culture media helps you to make difference between different types of bacteria. And here is a question. How do you think, how is it easier to find some specific information about bacteria by microscopy investigation or by bacteriological or cultivation? How do you think? Microscopy or bacteriology? Who is, you told bacteriology, who is for bacteriology? My group. Who is for microscopy? Anybody for microscopy? How is easier to find more features about bacteria? By microscope or by cultivation of bacteria? Nobody for microscope? No? Why? Because I gave you a clue. And my group also. So, apparently, sometimes you think, if it is microscope that shows us bacteria closer and we can see shape and so on, it is more precise, right? Apparently. And if you can see just culture media with some colonies, you don't see too much, apparently. But basically, if we put the question differently, how you can understand more about person by picture on Facebook or by its real life? Of course, you will say by their behavior in their real life. Because on social media, you can make fixation of the smear, and everybody thinks that everything is okay. Or you want everybody to think everything is fine in your life. Because you made fixation of smear. But when everyone is able to see you in real life, what you consume, what you produce, and it is not just about food and feces, yes? It is also about what you consume, what you are listening, what you are watching, and what you produce, which is the product you are giving in this life. Are you a useful person in this society or not? It is possible to find out just in case you can see the person in real life, not on Facebook, not on Instagram and stuff. This is why whenever students are asking if I have Instagram, I say I have YouTube. So please subscribe YouTube, not Instagram. Because on Instagram I'm nice, but on YouTube is product. Is a product I'm giving, and is a product you will need on the exam. So it makes me upset when I get more likes on Instagram than on YouTube. Please change it, okay? So if you can see the result of someone's activity, it is much more informative. Much more informative. This is how differential culture media helps us to find more about bacteria. One more interesting thing we should know, that some culture media, they combine two functions of selective culture media and differential culture media. For example, same McConkey we've been discussing in selective culture media. McConkey agar is used for detection of intestinal infections. So it is placed in selective culture media, and also you can observe it is also placed in differential culture media, this is the second one, because it helps not just to eliminate extra bacteria we don't need, but also by appearance, by color of colonies, or by color of culture media around colonies, changings of colors, McConkey agar, blood agar, triple sugar ion agar, also and others also are able to show us some features of bacteria. Because how it works, we take a pure culture, we use selective culture media, we obtain pure culture, we take differential culture media, we put it on these differential culture media, and this differential culture media will contain different nutrients and different chemicals. These nutrients are for different bacteria, because some bacteria will prefer as source of energy one kind of sugar, another bacteria will prefer another kind of sugar. Some bacteria will prefer glucose, another one lactose, another one sucrose, another one fructose, galactose, and so on. In dependence on these preferences of these bacteria, they will consume different types of sugars, and it depends what you consume, on this depends what you produce. So when bacteria will consume something, it will produce something, it will excrete something, and the results of bacterial metabolism will get outside in the culture media. So the chemical reaction will happen with metabolites of bacteria and the culture media reagents, chemicals, and the color of culture media will change in dependence on type of consumed sugar, or consumed or non-consumed iron, or you can see here example of triple sugar ion agar. This one is controlled. It is not affected. It is without bacteria. This one, you can observe that a little bit of bacteria, but it doesn't consume almost anything. A little bit of changing. Here, half is yellow because just one kind of sugar is consumed and one is not. Here, almost everything is yellow. That means almost all sugars are consumed. But here you can see the rupture of the culture media because of gas production. Some bacteria are able to produce gas as a result of their metabolism. When they produce gas, it destroys culture media. So you can see some bubbles and even rupture of the culture media. Excuse me. Some bacteria are able to consume iron also. As a result of metabolism, they will produce H2S, and it will give such a greenish but closer to black color, and will produce also some gas. So in dependence on all of these metabolic features, you will see different changings in culture media. So if we are coming back to microscopy, under the microscope you will see that almost all the bacteria that are causing intestinal infection are rod shape, gram-negative. Shigella, Salmonella, Escherichia, all of them are rod shape and gram-negative. So you will not be able even to make difference between different genuses, I'm not talking about species, under the microscope. Meanwhile, when we cultivate them on selective and differential culture media, you can detect even up to serovariants, biovariants, not talking about species, also species, but even sometimes up to some specific mutations that have different metabolism. This is what makes differential culture media extremely useful in bacteriology, in lab diagnosis. Also, of course, it is very important to mention specialized or special culture media because some bacteria are that capricious for the cultivation process that it is not possible to cultivate them on any kind of culture media. Moreover, often they are very slowly growing bacteria, and the mostly important example is Mycobacterium tuberculosis, Mycobacterium lepra, that are causing tuberculosis and leprosy. So here you can see an example of culture media, Levenstein-Jensen. It is here the first in the list. So here is an example of culture media we have for this type of bacteria. This bacteria is growing sometimes up to 6 weeks. Up to 6 weeks. Meanwhile, most of bacteria you can cultivate in 1-2 days. Mycobacterium tuberculosis will grow sometimes up to 40 days. A lot. So, at the same time, we'll put more Levenstein-Jensen culture media, and some of them with antibiotic dissolved inside, in order to check for antibiotical resistance, on which one will grow, on which one will not grow. Because we don't have too much time, 1 month to make cultivation, 1 month to make checkup for resistance and so on. We need time for treatment. Fortunately, we don't have just bacteriological investigation for now. So some investigation methods are much faster. This is what helps us to make faster diagnosis. And the last one is transport media. And if you pay attention how it was written till now, selective culture media, differential culture media, enrichment culture media, special culture media, and here transport media, without culture. Why? Yes, we can say transport culture media, but basically it is transport media. Why? We don't need to cultivate it. We need to transport it from patient to the lab. Sometimes the patient is coming to the lab. In this case, you take from patient and you put in the culture media and you make investigation. But sometimes in a hospital or in ambulance or anywhere, the facility of the hospital's lab doesn't permit you to make some specific examination, some specific checkups, or simply the lab doesn't work in the hospital, or simply we don't have a lab in the hospital. It also happens. So in this case we need something, because you will not take it in hand and to walk it through the entire city in order to bring it to the lab. You need to put it somewhere. And not somewhere, but in the place where the pathogen will be preserved in order to keep it until we bring it to the lab. Because some bacteria are that fragile that even to bring it few meters to the lab, it is too much. For example, Neisseria meningitidis. There is a rule to make culturing, to take the sample and to put it on the culture media near the bed of a patient. You come to the patient with culture media, you take a sample, you put it on culture media, and you put it in thermostat. So no transport media is provided for such bacteria. But when we are talking about other types of bacteria, that's enough just to put it in transport media and to bring it to the lab. No special temperature is required, no other special conditions, just transport media. So transport media can be applied for bacteria, for viruses, for different samples. It can be in different forms, it can be tubes, it can be in bottles, it can be on a Petri plate. Anyways, the mostly important thing about transport media, it preserves the pathogen from the patient to the lab. This is everything we needed to discuss today about culture media and bacteriology and bacterial metabolism. If you have any questions, you can ask.